Hello students, welcome to Bridge Academy. Today we'll be learning about osmosis in animal cells. In the previous lessons, we studied what osmosis is, and we also saw what happens in plant cells when placed in different solutions. We saw that plant cells have a rigid cell wall which prevents them from bursting. But animal cells do not have this rigid structure, and hence their problems are a little different. So let's start our discussion with the question, what happens to an animal cell if it gains too much water? Or what happens to an animal cell if it loses too much water? Let's find the answer together. The concentration of water in the blood is monitored by the brain. The brain sends signals to the kidneys to adjust the amount of water in the blood. Now, the kidneys are like little filters that clean your blood. They remove extra water from the blood and turn it into urine and send it out of the body. If there isn't enough water in the blood, the brain sends signals to the kidneys to conserve water and less urine is produced. By doing this, the kidneys keep the concentration of water in the blood within narrow limits. Since most of the animal cell is made of water, the concentration of water in the tissue fluid around the cell is almost the same as the concentration of water in the cell. As a result, there is no net flow of water into or out of the cells. But sometimes, due to certain factors, the concentration of water in the tissue fluid may become different from the concentration of water in the cell. What happens to the cell when this happens? Let's find out. In case the surrounding fluid has a high concentration of water that is hypotonic to the cell, water will move into the cell by osmosis. Water entering the cell will make the cell swell up. Animal cells have no cell wall and the membrane has little strength. If the cell membrane does not have enough strength to withstand the pressure created due to the entry of water, water would continue to enter and the cells will eventually burst. This process is called hemolysis of the cell. This can occur in red blood cells. Red blood cells in hypotonic solution become swollen and they burst, releasing hemoglobin into the blood plasma. Hemoglobin is the protein that carries oxygen in the red blood cells. When it is released into the blood plasma, it makes the blood appear red in color. Can you name such a condition? Yes, it is anemia. The symptoms of anemia include paleness of skin, shortness of breath, and tiredness. It occurs due to the deficiency of red blood cells or hemoglobin in the blood. One of the reasons for the deficiency of red blood cells could be the bursting of red blood cells due to hemolysis. Let us take an example of a person suffering from anemia. When he walks, he will feel tired and weak because his muscles do not get enough oxygen. This is because red blood cells carry oxygen to all the body parts. Now, what happens when an animal cell is placed in a hypertonic solution? A hypertonic solution has a low concentration of water. If the surrounding solution is hypertonic to the cell, water will pass out of the cell by osmosis and the cell will shrink. This process is called plasmolysis. Both hemolysis and plasmolysis are reversible processes provided it is done slowly. But if it is done quickly, it can damage the cell. For example, if someone drinks a large amount of distilled water very quickly, the water will move into the cells by osmosis. This will make the cells swell up and burst. This will also result in the death of the individual. Similarly, if someone does not drink water for a long time, the water from the cells will move out into the blood by osmosis. This will make the cells shrink and the person will die. To understand the importance of osmosis in living organisms, let us take an example of a potato. When a potato is kept in distilled water, it becomes soggy and even slimy. This is because water moves into the cells by osmosis. Since the potato cells have no cell wall, they burst and slimy gel comes out of the potato cells. When a potato is kept in a salt solution, it shrinks in size. This is because water moves out of the potato cells by osmosis. Thus, both too much water and too little water are harmful to the animal cells. In nature, the surrounding medium is never pure water. All natural water contains some dissolved substances. Hence, the problems of too much water or too little water rarely occur in nature. However, in the lab, we use pure water and solutions of different concentrations to study biological processes. Therefore, while studying biology in the lab, we need to keep in mind that too much water or too little water can be harmful to the cell. To summarize, we can say that animal cells do not have a rigid cell wall to withstand the pressure created by water. If the cell gains too much water, it may burst. This can occur when the cell is placed in a hypotonic solution. If the surrounding solution has a high concentration of water that is hypotonic to the cell, water will move into the cell by osmosis. Water entering the cell will make the cell swell up. Animal cells have no cell wall and the membrane has little strength. If the cell membrane does not have enough strength to withstand the pressure created due to the entry of water, water would continue to enter and the cells will eventually burst. This process is called hemolysis of the cell. If the surrounding solution has a low concentration of water that is hypertonic to the cell, water will pass out of the cell by osmosis and the cell will shrink. This process is called plasmolysis. We hope you have enjoyed this lesson. Do try to solve these questions. Thank you. Thank you.